like Angelica said, I'm Kara. I'm part of the clinical team with Microbiome Labs. I've been in clinical practice for the last 13 years and spent the good of last 9 years working with a large GI practice. I built an integrative functional program for them, and I live in Denver with my husband and my dog and my almost 17-month-old. So that's a little bit about me. I'm really excited to talk about the microbiome today, the world within. And I think Angelica said it so perfectly that even for us advanced practitioners, it's always good to go back to the foundation. It's always good to go back to really the basis of what does it all mean, right? Because that's how we really start to grow our knowledge. So I just want to take a moment to thank everyone for taking time out of their day. I know everyone is so busy, for joining us for this foundational series for our second portion. We're going to be diving deep into the world within, because we're no longer just a collection of what we thought we were, right? Organ systems connected by tissues and veins and arteries. But what we are, or a better representation of us, is a walking, talking rainforest. And so we're considered to be superorganisms, or a holobiont. Because we're made up of these 1,000 little ecosystems within that have to work together in order to prosper the health of the human host. Okay, so microbes are really at the origin of all life forms on this planet. And so some of you may be familiar with this diagram. There's lots of different iterations out there. It's the tree of life diagram. And so as you can see, I'm actually going to change to our laser pointer. So as you can see, microbes were the first inhabitants of the planet. They've played and will continue to play a pivotal role in shaping most living organisms that have followed after. We know that microbes are resilient. They've survived despite all forms that have followed. And they really have this very wise survival trait. We're going to talk about what those are as we move through our webinar today. But microbes were really quick to adapt to really all environments, very harsh environments, very extreme environments. And as we know that, and we can see here in this diagram, that bacteria were really the first and most successful form of life on this planet. Bacteria have been around the longest. They're the most successful, they're the most numerous, they're the most diverse and ubiquitous. They exist, everywhere from the bottom of the ocean all the way up to space. But they exist very deep within us, and they prove to be a very necessary and important aspect to human life. So maybe the most important ecosystem of them all is the human microbiome. So I want to first take some time to get some definitions out of the way. We often see these terms microbiome and microbiota used interchangeably, but there are definite differences between these two terms. So microbiome refers to the collection of genomes from all the microorganisms in the environment, which includes not only the community of microorganisms, but also the microbial structural elements, metabolites, environmental conditions, whereas microbiota describes the living microorganisms, so bacteria, archaea, eukaryotes, viruses, that are present in defined environments. And so throughout the body, there can be these niche environments, and you may see higher concentrations of specific microbes in one environment than you would see in others. And these niche environments are really important for microbes to be able to grow happily together in order to maintain the healthy ecosystem that in each site that they inhabit. And so we can see different environments from the oral microbiome to the gut microbiome to the skin microbiome. So human microbiota is estimated to be around 10 to the 13th to 10 to the 14th microbial cells. So that's around 1 to 1.3 microbial cells to human cells ratio. And this number you'll see as you dig through microbiome research, it's often, you'll see lots of different numbers out there. We're around 39 trillion bacteria in and on the human body. This weighs roughly the size of a human brain. But you'll see scientists often debating, whether we're more microbes than we are human cells. And you'll see these numbers, like I said, changing and floating around anywhere from 9% to 10% or more. But what is accepted and what's agreed upon across the scientific community is that we're mostly microbes. And that's why we're considered to be holobionts, that we're considered these meta-organisms. And that's because our microbes have around 3.3 million unique microbial genes. And so that's 150 times more genes than what we as humans have the capacity to do. And the capacity of the microbes actually far exceeds even, what our liver does, which, arguably is the most important organ in the body. So this is why we like to think of the human microbiome as a hidden organ, because it essentially plays, a pivotal role in virtually all physiological processes, from aging to digestion to the immune system to modulation of the central nervous system, our cognitive ability. And this is really what makes the human microbiome an essential organ in the human body, because without it, the human body just can't function. And so with the available technology that now exists, we can better define the intricate interplay between our microbiota and host health. And this was all really initially kicked off back in 2007, 2008 with the launch of the Human Microbiome Project. And this really launched a new wave of research, microbiome research, that has just exploded over the last 20 years. And that's really in part due to the ability to understand our microbial communities better. And this is all really accomplished with DNA-based microbiome studies. So I apologize, I have a little cold today, so I'm just gonna wipe my nose, and I'm gonna try not to do that too often. So DNA-based microbiome studies typically can be characterized as either targeted amplicon studies, 16S amplicon studies, or newer wave of technology being what's known as shotgun metagenomics. And this is, if any of you are familiar with our biome effects testing, this is what's done through whole genome sequencing. So 16S amplicon is primarily giving informative information via the taxonomical information. So taxonomic being who is in the gut or who is in the microbiome, while shotgun metagenomics is looking at the entire metagenomic sequencing or whole genome. And that's an approach that can also be used to study the functional potential of the community, not just who is in the gut, but what are they actually doing there. And so that's, if you're familiar with our biome effects test, that's what we're looking at, is who is in the microbiome and then what are the functional capabilities that they're actually performing. So this enables us to look, again, a little bit deeper into the microbiome, and all of this research and studying has provided, the basis and knowledge of, different compositions of microbiomes, and not only how it correlates, to overall health and well-being, but how it correlates with different health outcomes and conditions. So we can see now microbial profiles that for someone who may be, having active Crohn's versus someone in remission. And so we understand a little bit more about what are the microbial communities that take place, and are they performing those necessary functions. And so with all this technology and the explosion of research that's followed, there have been major advancements in understanding the collective human microflora and the communities that make up the various ecosystems within, because as we mentioned before, there's distinct microbial communities within the gut versus the oral cavity, in these varying environments that differ from one another. So we have four main phyla, and many of you are probably familiar with these. We have Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria. So we're going to talk a little bit more about these as we move on. But the microbial communities within our bodies, these are highly personalized. They're considered to be as unique to each individual as their own fingerprint. And all of these microorganisms work together and strive for balance within each of these ecosystems. And as we know, the gut microbiome is the largest and most diverse microbiome in the human body. It makes up about 70% of all microbes found in the body. Okay. So I'm just going to take us all back to biology class just for one very quick slide, just because it's always good to review taxonomic hierarchy or taxonomic rank. So I've already mentioned phylum. So phylum is a much larger high-level taxonomic classification. So in the example of bacteria, phyla would be Bacteroidetes, or an example of Homo sapiens, humans, right? We have phyla being Chordata. So as we get further down and further along on the taxonomic hierarchy, we get more and more specific, with strain being the most specific. So example in Homo sapiens is me, Kara Siedman, because I am very different and unique than everyone else sitting out there. And so in the bacteria realm, this is so important, because understanding species and strain can really be the difference between something that is commensal and healthy versus a bacteria that could potentially be pathogenic or opportunistic. So let's meet the gut microbiome. I'm going to take a moment just to review. This is something that you went over if you joined us for that first foundation series that focused on the digestive tract. So the lumen, right? If you remember, this is where the gut microbiome resides. It's technically still considered to be outside the body, but it's the innermost part of our intestines. This is where bacteria, bacterial products reside. And then we have the outermost mucus layer. This acts as that physical barrier, right? It protects what's happening in the lumen from getting further down into the epithelium. And so that's then reinforced further by biochemically with antimicrobial peptides and antibodies like immunoglobulin A. And then we have our intestinal epithelial cells. And this is what forms that single cell layer of protection, which is lined with those intraepithelial lymphocytes. And so these were mentioned. We'll talk a little bit more about these as we move on. And then within the intestinal crypt, we have our goblet cells, which help to increase that mucus layer, produce that MUC2 gene to increase that mucus layer. We also have our tight junctions, which we mentioned in our last foundational series. And then beyond that, we have what's known as the lamina propria. And this is densely populated with immune cells. This is where our innate and adaptive immune system resides. So this right now we're looking at, good question. This is the large intestine. And so the small intestine looks a little different. We know we don't have as thick of a mucus layer. Okay. Okay, let's go deep into the microbiome now. So our gut microbiome is the largest, it's the most complex, and it's the most diverse microbial ecosystem in the human body. So over 70%, as we mentioned, of the microbiota lives within the gastrointestinal tract and lives in this mutually beneficial relationship with its host. And we can see the increase of that bacteria steadily increasing from the gastric lumen to the small intestines, with the most concentration being in maximum concentration being in the colon and the rectum. And so this is something we actually often get asked is there bacteria in the small intestines? Well, yes, there is. It's just not as concentrated. And that's because there's a lack of limited presence of oxygen, bile acids that are there, antimicrobial peptides that are more concentrated in the small intestines. Whereas in the large intestines, this is a much more favorable area because there's lower concentration of antimicrobials. There's also a much, much slower transit time in the large intestines. And so we see that greatest concentration being in the large intestines. And so our normal gut microbial community, what we call our commensals, they don't cause any harm, and our commensals work harmoniously with the host to provide those many essential functions, which we're going to talk a lot more about. So the gut microbiota in healthy individuals is populated with those same four major phyla, right? Our Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria, where Firmicutes and Bacteroidetes should make up to 90% of our gut microbiota. And the gut microbiota of each individual becomes more unique as we get further down from genus and species level. Faecalibacterium or Faecalibacterium prausnitzii, this is one of the most common species in the healthy adult, which we'll talk a little bit more about as we continue on. So just to go over some more definitions, lots of definitions today, but microbiome structure. This refers to the number and types of microbes that are present. This is also known as microbial diversity, whereas microbiome function refers to the metabolic activities and the end products that are essential in maintaining a balanced ecosystem. And one of the things we like to say, you'll often hear a lot of other people in the microbiome world say this, that structure begets function. So structure brings on function. It reminds me of something someone told me with my when I first had my baby, that sleep begets sleep. So sleep brings on sleep, right? So structure brings on function. So if structure brings on function, then that microbial diversity, this is a strong indicator of the overall health, or more specifically, we like to think of it as a very key characteristic of having a healthy microbiome. So many of you are familiar with this schematic, but as you start to read about microbiome research, as you start to dig into the research, you'll read and you'll see that there really isn't a consensus about a definition, right? I like my definitions, but a definition of like, what is a healthy microbiome? And this is really because of the high, inter-individual variability that exists within each microbiome. But from the research, we've really gathered an understanding of key characteristics that are necessary to maintain the structure and function of the microbiome. So first, we obviously talk about good microbiota diversity. So having both abundance of species, so lots of species, the number of species, but also the richness or evenness of species. We also want to see a low level of pathogens. And so if we have that strong and good microbiota diversity, then this is essential as it reinforces that strong mucosal barrier. And that strong mucosal barrier is so key because it prevents what's happening in the lumen, what's happening in the microbiome, from what's happening in our immune system. And our mucosal barrier is highly dependent on overall diversity. So if we have that strong barrier, we have those strong tight junctions, as we talked a lot about in that first session. And so when these healthy conditions exist, the gut microbiome exhibits stability and resilience, as well as a symbiotic interaction with the host. Okay, so this was just a study that really looked at what we're talking about, which is diversity. So this was a study that looked at alpha diversity, which is a predictive index, or which is an index for diversity as a predictive index for health status. And so alpha diversity, or you'll often see it displayed as what's known as the Shannon index. This is a marker for species abundance. So how many and what's the evenness of species? Because you can have maybe 100 species, but if you only have 1 or 2 species that are dominating the microbiome, there's not uniformity or evenness, and that's a sign of a loss of diversity. So it's not as rich. And so this study was basically showing that they found a higher Shannon index, a higher alpha diversity in healthy versus non-healthy subjects, and they compared it to various disease states. So we can see that varying diversities within various conditions. So in general, the studies and other microbiome research studies have shown that having greater microbial diversity and functional redundancy can actually provide resilience to those changes that our microbiome can encounter, and therefore that's associated with having greater beneficial impacts on the health of the host. So as we discussed in the very beginning of our webinar, bacteria being the very first inhabitants on Earth and will ultimately be the last, but what about with human life? So who's born first? Is it baby or microbiome? So our exposure to microbes actually begin in utero and are profoundly impacted and influenced by our maternal microbiome. And so in fact, the fetal gastrointestinal tract is initially sterile, and it's influenced by mode of delivery, as well as surrounding environments. So obviously through the vaginal canal or cesarean, but then our skin. So all of these are going to influence the start of the fetal microbiome. And around 6 months of age, the composition actually starts to shift, but you'll see that we still maintain a very low alpha diversity. So we still don't have a lot of different species. And around 14 to 16 months, that's where we'll start to see the diversity increase. And this is often because, we're starting to get more introduced to, solid food. And so we'll see the diversity of that microbiome, adjust to that. But interestingly enough, we know that microbiomes of breastfed babies actually look a little different than the microbiomes of formula-fed babies. But by the time around 18 months of age, the microbiomes actually become indistinguishable, which is to me actually quite interesting. And then around 3 years of age, the microbiota profile resembles that of an adult. So the development and lifespan of the intestinal microbiota is really impacted by lots of different exposures to lots of different things. And this will essentially have profound effects on the types and activities. And we can see as we start to get older, we start to lose that diversity again. So there's many different factors that influence microbial communities. And although the population can remain relatively stable over the course of a lifetime, it is highly affected and modulated by both internal factors. So from genetics to obviously external factors like our diet, maternal factors, maternal influence, right? We see a lot of studies being done on immune activation in the maternal microbiome, mode of feeding. These are all going to, have an impact on microbial communities. And this really, as a practitioner and as for those of us working in this realm, it offers, exciting possibilities of controlling, health and disease because we can have control over some of these external factors, what we like to call the exposome. So all these different influences or factors that influence our microbial communities, or what we call microbiome disruptors. But this is actually why there's a lot of challenges when it comes to microbial studies, because there's so many different factors that come into play. So let's start to get into really the bulk of our session today is, what are our microbes doing? So we understand that there's these compositions, but what's this mutualistic and symbiotic relationship that exists between the gut microbiota and host? And we know this is regulated and stabilized by a complex network of interactions that encompass, metabolic, immune, and neuroendocrine crosstalk. So it's the job of our microbiota to essentially protect the host. Our gut microbiota are responsible for more than 98% of the genetic activity of the host. And this is why we consider it to be the second genome for the human body. We consider this a lot, right? Because we also consider our microbiome as the second brain, right? So we know that it plays a huge impact on just about, every aspect of the body. So it's also, we also consider the gut microbiome as like an additional endocrine system for the human being, as it can produce, a wealth of different substances that are necessary for the activity and proper functioning of organs and systems. So our microbiota have various functions that span from metabolic functions to structural functions, as well as protective roles. And the metabolites that are produced by our microbiota, these act as signaling messengers. And so this is what we call our microbiota crosstalk. This is essentially what links our gut with other organ systems in the body. So let's delve first into different metabolic functions of our microbiota. So we know that complex carbohydrates, plant polysaccharides that are not digested by human enzymes in the upper gastrointestinal tract, so specifically in the small intestines, require fermentation by our intestinal microbiota. And this is a process known as saccharolytic fermentation. And at the end, the byproduct of this fermentation is what's known as short chain fatty acids. So these are the primary end products. So what's known as acetate, propionate, and butyrate. We know that our short chain fatty acids are important regulators of gut microbial ecology, as well as host physiology. They play a role in essentially all aspects of human health. And the amount of short chain fatty acids produced can actually vary according to the availability of fermentable carbohydrates that are present in the diet, as well as the environment. The intestinal environment itself can actually impact how much is produced. So available short chain fatty acids are readily taken up by the host, but they can actually reach different levels of systemic distribution. And this is often why stool measurements may not necessarily be an accurate representation or an accurate picture of total short chain fatty acid production, because ultimately it's the genetic ability for our microbes to produce these substrates, to produce these end products, these short chain fatty acids. Bacteroidetes, these are our primary producers of acetate and propionate. Firmicutes, which we know lots of pathogens live there, but lots of important species live there as well. And these are primary butyrate producers. So short chain fatty acids have diverse physiological roles in the body and have an expansive role in the health of the host. We see that short chain fatty acids play a role in energy homeostasis, so the synthesis of certain gut hormones like glucagon-like peptide. This is in the news all the time now with everyone who's taking Ozempic and semaglutide. So again, our gut microbiota have the ability to produce this. So really always linking back to what's going on in the microbiome. We see our short chain fatty acids increasing leptin production, which is a hormone that helps to regulate energy balance by suppressing hunger, improving glucose tolerance and insulin sensitivity. Short chain fatty acids also have epigenetic activity as potent HDAC inhibitors. And this means that they can actually turn on and off certain genes that are involved in antimicrobial immunity and intestinal immune tolerance, and immune tolerance to bacteria, as well as to antigens and dietary proteins. They play a role in intestinal gluconeogenesis, lipogenesis, suppression of lipoprotein lipase inhibitor in the intestinal epithelium, and also have very strong immunomodulatory effects. The short chain fatty acids can act as signaling molecules that activate either pro or anti-inflammatory cascades. Our immune cells, our dendritic cells, and our regulatory T cells both have short chain fatty acid receptors on their cell surfaces, which helps to promote that immune tolerance. And we're going to talk a lot more about that. I will make sure that I get to everyone's questions at the end, just so we can keep on going. So please keep putting questions in the Q&A box and not the chat box, okay? Thank you. Forgot to mention that earlier on. So one of our most important short chain fatty acids, which is butyrate, this is the major energy source for our colonocytes, right? Colonocytes are a special type of epithelial cell. And as you can see in this picture, I won't go through it in so much depth, but the benefits of butyrate are far reaching. And they play a role in just about every system of our body, from our nervous system to obviously the gut to adipose tissue, right? So they're playing a role in just about every system of the body, all the way to the brain. So there's lots of bacteria that can produce short chain fatty acids, and there's also a special group of species, or a very important group of species, that we know are very strong producers of our short chain fatty acids. And so this is what's known as keystone species. And keystone species are an important ecological concept that is, that every complex ecosystem has. And essentially every complex ecosystem is structured by these very few important species, and that's what's been dubbed as keystone species. So these are beneficial bacteria that have a disproportionately large effect on both their habitat and the status of other microbial communities of the gut. So they don't take up a huge amount of real estate. In fact, our keystone species really only make up about 1 to 3%, sometimes 5%, but they have a profound impact on the overall health of the microbiome. And these protective bacteria produce short-chain fatty acids that can act as food for other bacterial species. But they also help to control the acidity and the inflammatory tone of the gut. So examples of these keystone species, what we have are primary degraders. So our resistant starches, our complex carbohydrates, are going to initially get fermented by primary degraders. These are bifidobacterium species and ruminococcus species. And then, resistant starches, cellulose, right? They get broken down into those oligosaccharides. And then our secondary degraders, so some of these more common keystone species that we know, Akkermansia and Faecalibacterium prausnitzii, Eubacterium rectal, Roseburia, these will break down those oligosaccharides and convert them to butyrate. This is why we call, if you've ever heard any of us in any of our webinars, we like to talk about the keystone species as being the microbiome, Instagram influencers of the microbiome, because they play a huge role. And these keystone species have, not only unique, but very crucial role in the microbial communities, but their removal. So as we lose diversity of these keystone species, this can actually cause a dramatic shift in not just microbiome structure, so the diversity of our microbiota, but also the functioning of our microbiota. So keystone species create an environment that's essentially unfriendly to our pathogens. They support other beneficial microbes by producing those short chain fatty acids. And so low levels, and this is what this study is actually showing, but low levels of keystone species has been associated with lots of different chronic conditions like obesity and diabetes and other cardiometabolic conditions, from GI issues like IBS and IBD, allergies and asthma, even chronic fatigue syndrome. So these become really important when we talk about the microbiome and of course the functioning of producing these short chain fatty acids. So another very important key function of our microbes is the ability to actually synthesize vitamins, primarily our B vitamins and vitamin K2. And 40 to 65% of the bacteria actually have the capacity to do this. So I know there's lots of words. It's really just to show you kind of the varying, I'm not going to go through all this, but you all will get our slides. But the different bacteria that produce these different vitamins. And this is important because a diverse and balanced gut microbiome can potentially make up for dietary insufficiencies. But we have to keep in mind that when we're looking at the synthesis of vitamins, it's not a measurement of sufficiency or deficiency. So we're not looking at like serum measurements. It's the genetic capacity or the ability of these different microbes that can produce these different vitamins. So for example, if we look at something like folate, vitamin B9, our microbiota can actually contribute up to 37% of the daily recommended intake. And so this is important because if we are running, stool tests, or we're looking at the genetic function of our microbes, and if these vitamins are low, we can actually, or let's say intake is low, we can actually rely on our bacteria to minimize that vitamin disparity. I also love to make note of vitamin K2, which can only be produced by bacteria. We, of course, get it in some fermented foods, but it's not so rich in the diet. And so if we don't have diversity, if we have changes in the microbiome, then essentially we're going to see changes in the ability of these vitamins to be synthesized. And again, when intake of these vitamins are low, I think this is important, when intake of these vitamins are low, especially in the diet, like vitamin K2, then what our bacteria are producing endogenously minimizes that vitamin disparity. Okay, another key function has to do with bile acid metabolism. I could spend, hours and hours and hours going into more depth on these functions. So hopefully this kind of sparks everyone's interest to start to dive a little bit deeper. But this is a really key function of our microbiota. So in the gut, our primary bile acids like cholic acid and chenodeoxycholic acid, they're synthesized from cholesterol in the liver. And then they're transformed by our gut microbiota to form what's known as secondary bile acids. Secondary bile acids like DCA, deoxycholic acid, and LCA, lipocholic acid. So primary bile acids in the intestinal tract can be reabsorbed by the host, by humans, and delivered back to the liver. And this is what helps to maintain our bile acid pool. But in the terminal ileum, as you see over here in the colon, the primary bile acids that escape that enterohepatic circulation will then be transformed by gut microbiota into secondary bile acids. And this is mainly through bile salt hydrolase, an enzyme produced by the intestinal microbiota that essentially removes glycine and taurine from conjugated bile acids to make secondary bile acids. So in humans, 95% of bile acids are reabsorbed into the distal ileum, and then the 5% unabsorbed primary bile acids are then bioconverted or deconjugated to our secondary bile acids, that's DCA and LCA, and that's done via the BSH, that enzyme. And that enzyme is secreted by several different types of bacteria, and you can see some examples here, different types of Clostridium. So Clostridium scindens is a keystone species. In fact, we know it's a bile acid metabolizer. And then the rest is excreted in the stool and urine. This is what's known as passive transport. So bile acids can actually shape the gut microbiota community, and having that BSH activity actually enhances bile tolerance. So bacteria use this to survive in the gut. And having a low ratio of primary bile acids to secondary bile acids is actually associated with dysbiosis, or an imbalance in our ecosystem. Our gut microbiota can also metabolize amino acids into specific microbial metabolites. And so while there are benefits to these metabolites, many of them in large quantities can actually play a key role in many different microbiome related issues, like having increased intestinal permeability, inflammation, even neuroinflammation. So amino acid fermentation typically occurs, this is what's known as proteolytic fermentation. So this typically occurs in less acidic conditions, also when there's insufficient fiber intake. So this means that the overconsumption of protein can actually increase the risk of producing more potentially inflammatory compounds faster than they're actually able to be excreted from the system. So no, I'm not saying that protein is inflammatory, but these shifts to proteolytic fermentation, and then the subsequent release of these pro-inflammatory metabolites like histamine and P-cresol and polyamines and ammonia, means that potentially sufficient digestion of protein isn't happening. And so digestion of protein is essential. So when dietary fiber is like your resistant starches, your prebiotic fibers, when these are available to the gut microbiome, when they're sufficient in the diet, the majority of proteolytic fermentation byproducts will actually produce short chain fatty acids. But when there's shifts when those shifts occur, and the result are these more harmful byproducts. So when we shift more towards a proteolytic fermentation, then the abundance of these byproducts can actually become more harmful and toxic to the body. And this is actually a sign of dysbiosis and can actually be potentially signs of digestive insufficiency. Maybe protein didn't get properly digested or denatured in the stomach prior to working its way through the intestines. So as a result, many of our pathogens and our opportunistic bacteria, they come in, they decrease the acidity within the GI environment, and this creates an environment that's essentially more favorable for proteolytic fermentation to take place. So it isn't always because there's dietary discrepancies, but it could actually be a sign of dysbiosis and an imbalance in our microbial ecosystem. But when it comes to the diet and what we do know about these shifts is that it really highlights the need, and really the importance of fiber and balance in our macronutrient intake. So I know a lot of people are like, oh no, what about, carnivore style diets? And as a clinician, if I have someone who's following, a high protein diet and they feel amazing, most often I'm not seeing them then, right? Because they're not coming to seek me out. But it just calls for need to still support the microbiome and to support the function of our microbes. So I just make sure then that I'm either supplementing with functional fibers or providing prebiotics so that we're really giving our microbes the food that they prefer. So we're going to dive a little bit into specific amino acids and their metabolites. So the first being tryptophan, an indole metabolite. So tryptophan is an essential amino acid that's solely obtained from the diet. It's entirely dependent on dietary intake as well as gut microbial production. So tryptophan is metabolized to various bioactive compounds, and these are through 3 different pathways. We have the kynurenine pathway, that's via our immune cells that are in the liver and the periphery, via intestinal lumen, so enterochromaffin cells, as well as via gut bacteria, our indole pathway. And our gut microbiota transform tryptophan into these various tryptophan and indole metabolites, and these essentially act as what's known as ligands for AHR, aryl hydrocarbon receptors. And that's a protein that essentially triggers a wide range of effects like modulation of the metabolism, immunity, psychosocial, so play a huge impact in everything from drug resistance to biofilm formation and virulence factors, as well as intestinal barrier function. So we'll talk about a lot more about those. Choline metabolites, so the production of what's known as trimethylamine. This is produced by the gut microbiome from lecithin, phosphatidylcholine, choline, and L-carnitine rich foods, and these are commonly found in red meat, full-fat dairy, eggs, and fish. And so this depends on our gut microbiota for trimethylamine is oxidized in the liver to TMAO, trimethylamine nitric oxide, which this has been shown to be positively correlated and associated with an increased risk of atherosclerosis and cardiovascular events. We see in the research that higher microbial diversity has actually been shown to decrease TMAO. So again, going back to the importance of that diversity, and excess TMAO production is a sign of gut dysfunction. And so it's having excess bacteria that is capable of producing TMAO. TMAO can actually inhibit bile acid synthesis, promote inflammation, thrombosis, again, affecting a lot of cardiovascular events. We see TMAO producers primarily coming from Clostridiaceae, Peptostreptococciaceae, so more proteobacteria, which if you recall, proteobacteria being one of our major phylas, but proteobacteria tends to contain a lot of our pathogenic bacteria and bacteria that contain a specific bacterial endotoxin known as LPS, which we'll learn a lot more about if you don't already know. So a very small proportion of total dietary fat actually reaches the colon. So microorganisms in the gut are known, actually less than 5%, but microorganisms in the gut are known to possess lipases, which can actually degrade triglycerides and phospholipids. So there's only a few metabolites that are produced that have lipophilic characteristics, and that means that they're actually able to pass through the epithelial barrier and directly impact host metabolism. So gut microbiota composition and its derived lipids can actually impact the host metabolic state by altering even plasma lipid levels. So we'll see here LPS being a bacterial endotoxin. So increased gut permeability, this can actually facilitate the translocation of LPS over that intestinal epithelium. LPS, if you've listened to any microbiome webinars, we talk a lot about LPS. This can induce immune activation and inflammation through what's known as TLR4 complexes. These are found in our immune cells. We'll talk more about these. And that can actually result in metabolic disruptions. But interestingly enough, and what studies have shown is that HDL may actually neutralize the toxic effects of LPS. Our bile acids are also important. These regulate metabolism by binding to what's known as FXR and TGR5 in several different tissues. We talked already about choline and L-carnitine metabolites. This is potentially a marker for atherosclerosis, that TMAO. And then, of course, our short-chain fatty acids stimulate incretin hormone production, supply the host with energy, improve peripheral tissue metabolism. And there's actually been some studies done that have shown that microbiota composition actually differs in diets that are high in saturated fat compared to diets that are high in our omega-3 fatty acids. And they found that saturated fat diets, or diets that are higher in saturated fat, actually have lower abundance of our beneficial and keystone species, so Akkermansia and Lactobacillus and Bifidobacterium. Xenobiotic metabolism. So our gut microbiota can biotransform xenobiotics. Xenobiotics are any substance that's essentially foreign to the human body. We have exogenous xenobiotics. These are things that are found in the diet, medications, environmental toxins, and then endogenous, which are synthesized within the body are produced as metabolites from different biological processes that take place. And we now can better understand that the gut microbiome can actually significantly disrupt or even accelerate the pharmacokinetics of xenobiotics. That's a tongue tie. And this can affect metabolism and the bioavailability of certain medications and drugs. And this is either through direct or indirect mechanisms. So direct, we know our gut microbiota can directly metabolize xenobiotics into active, inactive, or even toxic metabolites. Xenobiotics may also shape the composition of the gut microbiota, and that is through antimicrobial activity or selective growth. So an example of that is the nearly 3,000 predicted cytochrome P450 enzymes that are present in bacteria compared to the only 57 in humans. And then there's indirect mechanisms where our gut microbiota may actually metabolize oral drugs and dietary compounds prior to their absorption, and this essentially affects that first pass metabolism. And this will ultimately decrease their concentration before reaching systemic circulation and before reaching those targeted tissues. So microbial metabolism of pharmaceuticals can actually lead to either their activation. An example, a medication that this happens with is like sulfasalazine that we see. Inactivation, we see this with like methotrexate and digoxin, or can actually result in the production of toxic metabolites. And we see this with certain anti-cancer medications. And so microbial metabolism of these drugs can have an impact on their side effects, as well as drug therapy through the interference with the host pathways that are involved in drug detoxification. Okay, so that concludes the metabolic functions. I'm gonna move on, so since I'm running behind a little bit. But now moving on to really one of the most surprising and probably one of the most, really important areas that have been observed in microbiome research is that our microbiota, our bacteria, don't simply just evade the immune system, but there's a complex interplay that exists between the local microbiome, our intestinal epithelium, our resident immune cells, which are all pivotal in maintaining gastrointestinal homeostasis. So our immune system development correlates with the development of the microbiome. And so our gut-associated lymphoid tissue, you heard about this in the first series. This is a component of mucosa-associated lymphoid tissue and makes up about 70% of the immune system. This is why we say 70% of the immune system is contained in the GI tract. And this is comprised of our lamina propria, intraepithelial lymphocytes, mesenteric lymph nodes, Peyer's patches. This is everything that you guys learned in your first section. And so the immune system, right, is not innately hardwired to tell the difference between something that's a harmless bacteria versus a threatening pathogen. It's the microbe that actually makes the distinction clear. So the role of our gut is to manage the immune response to the massive antigen exposure that's experienced by the gut, while also maintaining, potent adaptive immune responses to protect the host from mucosal pathogens. So our immune system is composed of both innate and adaptive components, and this has, the ability to adapt and respond to highly diverse challenges. So I always like to say that, our gut microbiome is the eyes and ears of the immune system. And in turn, the microbiota can promote and actually calibrate all aspects of the immune system. Certain microorganisms even help to develop our immune cells. So let's talk about innate immunity. This plays a significant role in gut because it provides a more rapid response. So innate immunity is our first line of defense against germs and pathogens that are entering the body. And it responds in the same way to all germs and foreign substances, which is why it's sometimes referred to as like a non-specific immune system. It acts very quickly. So our immune system, our innate immune system has really only a limited power to stop germs from spreading throughout the body. And our commensals can actually modulate the function of our innate immune cells. So we have our antigen presenting cells, like our macrophages and dendritic cells. And these are important because our tissue resident innate cells, our innate immune cells that phagocytotize and secrete pro-inflammatory cytokines. And so this is important when something is getting presented or an antigen is being presented to the immune system. We also have our dendritic cells, which is responsible for that presentation of antigen to T cells. And these are recruited to the gut mucosa by our commensal organisms. So our microbiome helps these cells by expressing what's known as toll-like receptors. And these TLRs actually neutralize the immune response to offer tolerance. This is how we start to build immune tolerance. Our microbiome also goes as far as producing ATP, which we know is our form of energy to help these cells differentiate and function. Our natural killer cells, this is a major part of the innate immune system, and they specialize in identifying cells that are infected by a virus and essentially helps to eliminate them. So having a dysfunction in our natural killer cells, an individual would face chronic consistent infections, and our microbiota can actually stimulate the production of these cells. Microbiota also affects the potency of these cells. And then we have our intestinal epithelial cells, and these are things like your goblet cells that produce our mucus, enterocytes, which are involved in nutrient absorption, our enteroendocrine cells, which secrete hormones. These release key antimicrobials to protect our barriers. An example is intestinal alkaline phosphatase. This is an enzyme that actually helps to dephosphorylate and actually detoxify LPS, so really essential. Our intestinal epithelial cells release chemokines and cytokines to recruit immune cells to the location that's needed. And the microbiota also stimulate the intestinal epithelial cells to release these antimicrobials and chemical messengers. And we have other immune cells like mast cells as well. So when we look at the microbiome and innate immune system, our gut microbiome helps to educate the immune system to actually differentiate between what's harmful versus what's beneficial. And certain bacteria in the gut produce certain substances to actually help prevent excessive inflammation. So our microbiome can influence our immune system's responses. It can actually help prime the immune system to respond more effectively to infections and antigens. So having a diverse and healthy microbiome is associated with a well-functioning immune system, reducing the risk of chronic conditions and allergies and immune-mediated disorders. So our adaptive immunity. So unlike the innate immune system, which is pre-programmed to react to common broad categories of pathogens, our adaptive immune system is highly specific to each particular pathogen the body has encountered. So adaptive immunity actually creates what's known as immunological memory after an initial response to a specific pathogen, ultimately leads to enhanced response to potentially future encounters with that pathogen. So antibodies play a critical part of the adaptive immune system, and adaptive immunity can provide more long-lasting protection, sometimes up to, for a person's lifetime. So our adaptive immune system is made up of T cells as well as B cells. And so our T cells contribute to immune memory. They're produced in our bone marrow, and then they move to the thymus. That's where they get the T in their name. And our Treg cells, these are suppressor cells that downregulate the inflammatory response. And then we have our T helper cells, so Th1, 2, Th17, and as well as our T follicle cells. These are what's known as T effector cells, and they essentially use chemical messengers to activate other immune system cells in order to start the adaptive immune process. And so they can actually detect cells that are infected by viruses or tumor cells. They can destroy them. And then some T helper cells actually become memory T cells after the infection has been defeated. And so they can actually remember which germs were defeated, so that they can, have that memory if they get re-encountered with them. So our B cells, these are antibody secreting cells. They get their name from bone marrow, so the B from bone marrow. And like T cells, there's many different kinds of B cells that match particular germs. So our gut-associated B cells primarily secrete IgA. This is the antibody that's made in the highest concentration. They originate in our pairs patches. And so when T helper cells actually contact B cells that match the same germs that they do, this activates the B cells to multiply and essentially transform themselves into plasma cells. And the plasma cells quickly produce that large amount of antibodies to release into the blood. And interestingly enough, our body can actually make up to 7 grams of IgA a day. So the amount of B cells and their potency is directly controlled by our commensal bacteria. IgA, unlike IgM, actually has low memory, and so mostly recognize the current crop of commensals and invading organisms, so it requires constant stimulation and upregulation to provide those new IDs and protection. Low microbiota diversity, low microbial exposure, low antigenic species in our environment leads to low levels of IgA production. So microbiome and the adaptive immune system. So the gut microbiota play a significant role in shaping and modulating the host's adaptive immune system. And this interaction involves several mechanisms. So one being tolerance, which is establishing immune tolerance to ensure the immune system recognizes harmless antigens and doesn't overreact to them. That's essentially what reduces oral tolerance and why we can often start experiencing food sensitivities. Regulation of T cell development. So gut microbiota are the most prominent influence from the environment on differentiation of our T cells, our Th17 cells, and promoting the differentiation of Treg cells. That's necessary to maintain immune homeostasis. And then also influence on antibody production. As we mentioned, our microbiota contribute to the development and differentiation of our B cells, which produce antibodies like IgA. So just highlighting a few of those metabolites that we've talked about that were discussed that have very clear functions in immunomodulation. So short chain fatty acids can increase our regulatory B cells, inhibit those HDACs, can upregulate beta cell metabolism, which increases the production of IgG and IgA. Our tryptophan metabolites help to regulate immunity and also help to upregulate anti-inflammatory responses. So one of the most important aspects of gut homeostasis is obviously the function of the intestinal barrier and the gut barrier and the ability to maintain that boundary between the lumen. So maintaining what's happening in the microbiome versus what's happening in the immune system. So maintaining a set pH, a strong mucosal layer, those strong tight junctions, as we mentioned earlier, is key to having a healthy microbiome that's both strong and resilient. So functional immune responses often modulate gut barrier integrity. So the fine tuning of our gut microbiota to evoke those necessary immune responses to alter the intestinal barrier function is actually very critical. And as you can see, various gut microbiota metabolites will modulate the intestinal barrier. So the intestinal barrier, which we've already talked about, that chemical barrier, the physical barrier, the immune barrier, we know short chain fatty acids can actually enhance the chemical barrier by stimulating the secretion of antimicrobial peptides, secretory IgA, which we just discussed, mucins to prevent harmful bacteria, short chain fatty acids, bile acids, indole derivatives. So all these metabolites that we've been discussing can enhance that physical barrier, increasing tight junction proteins. We also see short chain fatty acids and indole derivatives can cross that epithelial barrier and act on immune cells specifically to release the production or release anti-inflammatory cytokines like IL-10 and IL-22. So this is just a schematic showing the potential pathways that are responsible for microbial mediated enhancement of our gut barrier function. So healthy diets, again, are metabolized by our beneficial bacteria to produce various metabolites. And each of these microbial metabolites activate various pathways responsible for modulation of the gut barrier. And by supporting gut barrier function, then we essentially are supporting inflammation and preventing immune activation. Okay, so moving on, almost done here. So enteric nervous system, central nervous system development, we all have learned a lot about the gut-brain axis, and our microbiota and our commensal bacteria impact the gut-brain axis both locally with our intestinal cells and the enteric nervous system, and directly with the central nervous system through neuroendocrine and different metabolic pathways. And so our brain actually modulates gut function through the HPA axis, as well as our autonomic nervous system, and our gut can modulate central nervous system functions through involving a variety of microbiota-derived metabolites and products. So gut microbiota to brain, we can see that via different metabolites, our gut hormones, neuroactive substances, and then brain to gut microbiota. And again, this is via the HPA axis and enteric autonomic nervous system, and this will affect motility. And they've even done studies on germ-free mice that have shown that bacterial colonization of the microbiome is really central to the development and maturation of both the enteric nervous system and central nervous system. And they found that neurochemical and behavioral effects in vagotomized mice, that they were actually not present. So that really identified the vagus nerve as a major modulatory communication pathway between the microbiota and the brain. And so our gut microbiota produce certain neuroactive metabolites like neurotransmitters or their precursors, which can affect the concentration of neurotransmitters, their precursors or both in the brain. And so this ultimately suggests that neurotransmitter synthesis pathway in the intestine might directly or indirectly affect neuronal activity and cognitive functions of the brain. So our bacteria produce enzymes that facilitate the synthesis of neurotransmitters or their precursors, and these precursors can pass through the blood-brain barrier and participate in the synthesis of various neurotransmitters. And we see this with a lot of our metabolites, like even short-chain fatty acid deficiency being associated with neuroinflammation and conditions related to neuroinflammation. Okay, so we made it to really the final, key function of our microbiota, which is along the lines of the protective function. This is what's known as colonization resistance. And this is where indigenous or commensal microbiota provide protection to the host against colonization of pathogenic invaders and provide prevention against the overgrowth of pathogenic microbiota, so or what we call pathobionts. So during perturbations or changes to the microbiome, there's a decrease in the dominant microbiota members, reducing the colonization resistance capacity. And that allows for more opportunistic pathogenic strains to actually invade or colonize these empty homes. And this can essentially lead to occurrence and infection. This is what happens with like a C. diff infection. So commensal bacteria actually regulate the population and activity of pathogenic bacteria via direct or indirect ways to maintain normal, healthy gut microbial composition. So direct interaction between microbiota and pathogens and competing for shared space. This is via the production of antimicrobial molecules, bacteriocins, secondary bile acids, change in the GI environment, so altering the pH, nutrient composition, this is what's known as quorum sensing, and of course through metabolite production. So again, our short chain fatty acids, our bile acids, secondary bile acids, indole. And then indirect mechanisms via the enhancement of host defense by microbiota to inhibit pathogens. And this is the induction of our immune system, which we just talked about. So just going through really quickly those different pathways. So we have direct mechanisms of colonization resistance. So commensal bacteria can consume dietary amino acids, sugars, and amino acid metals, as well as even oxygen, which can actually starve the pathogen of essential nutrients, what's known as quorum sensing. At the epithelial surface, bacteria can actually modify host glycolysis. I can't talk today, sorry. Glycosylation, which creates, sorry guys, I think this is the cold in my head, and use it as a nutrient for adhesion, which creates essentially a new environment for these pathogens. And then we can see that active antagonism, this is where symbionts or commensals can actually directly kill pathogens via antimicrobial substances or antimicrobial peptides. And this is not only preventing pathogenic infections by directly killing them, but it just limits that pathogen colonization. And then other inhibitory compounds like our short chain fatty acids, our metabolites that can suppress the expression of even virulence factors in pathogens. Bile acid metabolizers, like we talked about before, an example being Clostridium scindens, can convert our primary bile acids to secondary bile acids, which can inhibit pathogenic growth. And then indirect mechanisms, which is done by modulating the host environment to make the intestinal landscape just less susceptible to infection. So just making a less comfy home. So I won't go through all of this together, but we have the mucus layer, which we know is important. Fiber deprivation actually leads to the increase in mucus degrading bacteria. We know that butyrate promotes aerobic respiration in intestinal epithelial cells, and this will reduce oxygen levels in the gut. So that can actually limit the expansion of certain facultative anaerobic pathogens like our proteobacteria. And then of course, our immune mediated responses. So again, that activation of our innate and adaptive immune system. So it's really important that by, altering the composition, the functional capacity of our gut microbial community is impacted. And this affects things like colonization resistance, so the inability to protect against pathogens. And so this is what happens when our microbiota really lose that harmonious balance they strive for. We lose that function. And so our microbiome is really the foundation to health. And that's hopefully what we're taking away from this, that all the various functions that our microbiota can do. And that's everything, we've been discussing today, and how critical our microbiota are in promoting overall health and well-being. And so the factors, this exposome that we talked about earlier, this is really what your next foundational series is going to be focusing on, is how these microbiome disruptors, start to drive cracks into the microbiome, and what those disruptions essentially do, right? Because if our microbiota cannot perform those necessary functions, from immune regulation to nutrient metabolism to protection and everything else, that we discussed, we start to see those effects not only within the GI tract, but extraintestinally, virtually affecting every single part of the body. So in summary, our human microbiome, right, considered a virtual organ, it plays a key role in host physiology. It's the largest and most diverse ecosystem. Our gut microbiome is the largest and most diverse ecosystem. And keep in mind, structure is diversity, whereas function looks at the functional capability or capacity of our microbes to perform all the things necessary. And there's many different factors that can impact the microbiome and inhibit microbes from performing these necessary functions. And so that's what we're going to be talking about in your next session. So with that, thank you so much for joining me. I'm going to hopefully just take, if you have time, just to go through a few of the questions that are in the Q&A box. Hopefully my brain is working. So I think I already looked at the intestinal site. So that was the large intestine. When someone is on tube feeding, particularly a J tube, what probiotic might be helpful to prevent infections and provide? So this is hard. I mean, especially and you can even answer later, but if they're not taking anything by mouth, I'm always a little cautious with putting things through tube feeding only because they're in an immunocompromised state. Again, I'm kind of cautious on using probiotics in tube feeding. Are there studies related to gut health of infants based on how they are birthed? So absolutely. I mean, just do a PubMed search. There's so many studies looking at the gut microbiome and how this can potentially shape, that of an adult. So absolutely. How long after antibiotics would someone's microbiome function be restored? So it just depends. I mean, we can actually modulate the microbiome in a short period of time, but I think it depends on to what extent has diversity been impacted. And this is why, and this is going to be actually your fourth part of your foundation series, is how to actually restore gut health. And this is why it is so important to look at diversity. And I'm always a fan of running certain microbiome-based studies after antibiotics or after certain conditions, just to see how the microbiomes actually be impacted. But we can improve the microbiome really within a few weeks. I mean, we can. I think it just depends on how much, loss there's been. Okay, your thoughts about medium chain so medium chain fatty acids, so specifically like MCT oil, coconut oil, I'm assuming that. So I think it depends. There's been a lot of studies that look at saturated fat and actually coconut oil in particular, how this can actually increase the processing of LPS being a bacterial endotoxin that's found on our gram-negative, the outer cell membrane. And they've actually shown that coconut oil, what happens when we digest fat is we create these sort of lipid rafts, so it pulls in saturated fat to be processed and digested. And LPSs actually look very similar. So if there's dysbiosis, if there's damage or there's disruptions to our foundation, I might actually be cautious with MCT because we don't want them to drive inflammation and immune activation anymore, which is what happens when LPS can cross into the epithelium and get into the immune system. Yes, butyrate can be taken orally. In fact, I've used butyrate quite often, especially when I've done microbiome-based studies and I see that someone's butyrate's depleted. Yes, we want to build the microbiome environment, but if someone has irritable bowel disease or really bad diarrhea, I might add it in, even orally or as a suppository to try to help with those symptoms. Would elevated HDL, like 80, be an indication of having to fight off LPS? That's a good answer. I have to do some more research into that. And look, because sometimes having high HDL, depending on other genetic factors, can sometimes not, sometimes be an indication of cardiovascular risk. So that's actually good. I'll look into that. That's a good question. Let's see here. If you're okay, I'm going to take just like one or two more minutes. Yes, you're going to get the slides. So do we only get leaky in the large intestine? So that's a good question. Actually, there's been some recent studies that have come out in microbiome, in the research, that's shown that we can actually see intestinal permeability and we can see loss of that diversity actually taking place in the small intestines. And that's actually behind some of the conditions that we deal with like disorders of gut-brain interaction, and especially IBS. So looking at that loss of oral tolerance and that loss of immune tolerance, that's actually driving some of the changes that we're seeing in the microbiome. So yes, I think that some of the newer research is showing that yes, the colon has the largest environment, but we're seeing the same issues taking place in the small intestines. So with that, we're about 7 minutes over. I really, really hope that everyone enjoyed the webinar today. Everyone will get a copy of the recording as well as the slides, and I appreciate you listening to me even though I sound pretty nasally. And I hope everyone just has a really great rest of your day. And we look forward to having you join us in. We have another webinar coming up this month. And then our third foundation series in December, which, like I said, is going to go into dysbiosis and how this not only impacts the gut, but extraintestinally as well, that gut organ access. So thank you everyone for joining us. Have a great rest of your day.